Good morning. Welcome to session 3 on semi-automated curation tools and systems. We have a great set of speakers for you this morning or this afternoon or evening. These are people who've been leaders in creating approaches to organizing and making accessible the information in biomedical literature and other resources through scalable curation. Each speaker is going to have about 20 minutes to talk, but I will interrupt you or remind you at about 16 or 17 minutes so that we have time for questions. And I ask the audience, you are encouraged to enter questions in the Q&A. Please be specific about who you are directing the question to, and we will leave some time at the end to answer these questions. So with that, I will introduce Patrick Rusch, who is a professor at the HESSO University. That's the University of Applied Sciences and Arts of Western Switzerland. Patrick leads the text mining group of the Swiss Institute of Bioinformatics in Geneva, and that group develops and maintains different curation support services for the SIB databases, including UniProt. So Patrick, take it away as soon as your screen comes up. We've been having some bandwidth issues, so we may.
Okay. Do you hear me well?
Yes, but we don't see your screen.
Yeah, I know it's coming. We repeated this right before. I need first to enter the slideshow mode, and then I should try to share. So it should show up in the coming seconds.
Ah, okay, you're up.
Okay, thank you very much. So I'm going to talk about text mining and curation support tools. If I compare to what I was planning to show about 2 years ago, so things have moved slightly. So 2 years ago, I would have shown something about the different text mining services we were developing for a project called the Swiss Variant Interpretation Platform for Oncology, which has now expanded outside oncology, and which is probably useful for most genomics variants. So what happened is in the past 2 years can be summarized via what happened in the last 2 weeks, because we have had this bioinformatics paper that has been released, published in March. So, to make a long story short, what we started to do 2 years ago is a platform where clinicians in 5 university hospitals in Switzerland could submit requests for curation about variants, or eventually to submit a full VCF content. The idea was to screen through these variants and to gather all the information that could be available. Of course, locally, if we already have some curated content, but if needed, that would involve searching the literature and then develop a biocuration pipeline with the curators of UniProt to end up with actionable information. And the last piece of work, probably one of the most original, was that the clinician in these hospitals were supposed to provide a feedback. So the curation was supposed to be validated, whether a certain maybe variant could change the clinical synopsis or the treatment was supposed to be validated. So this is now something that is in production since December last year. But for the talk of today, we're going to focus on the literature search. So the objective is to develop a search engine to go into specific and relevant data collections. And I would say the main issue is probably a recall-oriented challenge, because most variants are in fact variants of unknown significance, therefore they are rare. So the general architecture is probably the same in most literature curation applications. So we start with the usual suspect, Medline, PubMed Central. We also work with supplementary data. And of course, for variants, we also are interested in clinical trials. And we can work with other types of resources like maybe biodiversity information. So this is what we have in the SIB text mining services and the literature services in particular. So for today, I'm going to focus on Medline and PubMed Central first. There will be one experiment we are going to report about the use of supplementary data. And for the clinical trials, they are in the tool, so you can test it online, but I'm not going to provide any evaluation of this. So what is the SIB literature services? Again, it's a sort of mirror of PubMed and that's it, together with a few specific collections, but it is the same type of ID. And within this mirror, we already pre-annotated several entities, about slightly more than 2 billion today. And we are adding new entities on a regular basis. Like today, we are working with biotic interactions and cell lines. The Celosaurus, which is maintained by SIB, is a new Elixir core data resource. So we are investing effort into this one. So the methods that we use are, state-of-the-art information retrieval methods. The basic query is a gene and a variant, and optionally could have a disease. Okay, so the strategy is pretty much the same in this type of application. So you look for all the papers you can get. You try to gather them using normalization, for disease and drugs, and expansion for instance, for variants. And at the end, you build a linear combination, which is hopefully optimal. So if I look at so for the normalization of the entities, we already showed what we have into SIB literature services. Now, if we look at the variant expansion, so we have a very broad expansion service, which is working at a protein, transcript, or genomic level. It is able to expand all the standard formats. Of course, we use dbSNP, HGVS, and the like, but we also are able to expand non-standard formats. And of course, in the literature, you may have relatively rich ways to describe a variant. So I'm now reaching the evaluation, which is probably the most important part. So we deployed several, let's say, experimental settings, and I'm going to go through the most important. So for the first one, which is the use of the system as a literature triage system to curate one or two variants. So this is what we call on-request curation, which is the one that is preferred by the clinician, by the way. So we use Trek benchmark to do that. And the system is relatively competitive. We were always in the top 2 or 3 at Trek. And the conclusion is that we are able to find the relevant papers in the top 5 with 2/3 of the time. What is interesting here is that when we use a relatively powerful variant synonym expansion system, the performance degrades. And the only explanation for this, even when we control for very, I would say, high quality variants, is probably that the benchmark is just not appropriate for such a task. So in the sense that the Trek curators did not look at the synonyms. The second evaluation is the use of the system as a variant ranking. So the idea is to provide dozens of or hundreds of variants as it is available in a VCF file, so which is the result of a variant calling output. In that case, we use a benchmark provided by the Swiss tumor board. So these 5 university hospitals have joined forces to have a common tumor board, and they provided us with some interesting data from 8 patients, but for a total of 756 variants, and each of them was marked up with, in a sense, interesting variants. So about 1 or 2 per patient, and the idea was to check whether we could top rank these very interesting variants. So the results are on the right. So out of the variants, the total variants that we work with, 2 of them could not be found at all in the literature. So which means about nearly 20% silence. And the others were always ranked in the top 1, 2, or 3. So the average number of variants per case was 94. So it's relatively non-trivial, I would say. But the third evaluation is a comparison between the system and LIDVAR. And for that one, we used a data set from BARCA Exchange, which is a subset of LOVD, comprising variants for BARCA1 and BARCA2, so about 800 of these variants. And on the right, you see the Venn diagrams of the results. So we significantly improved the recall per document, but also probably more importantly for the curators, we reduce the silence. It means all these queries which just did not find any match in the literature. Now, if we look at further down into this evaluation, we can look at more in detail into this subset where LIDVAR is doing better. So we selected randomly 10 queries, and the results are shown here. So of course, this is a manual evaluation, okay, and it could not be possible at large scale, but we can always sample a subset. So for 7 out of the 10 variants, this was simply LIDVAR was included into variants. And then for 3 out of 10, LIDVAR retrieved 6 documents that were not retrieved by our system. And then if we look at in detail why we did not find them, for 2 of them is because the variant is found in the reference section, and the reference section is not indexed by the tool. So the assumption that we discussed with the curators is that to have a mention in the bibliography is not sufficient to make a paper relevant. And for 4 out of 6, the variant is found in the article, but not the exact variant, just the position. This is coming from the fact that the RSID, so the dbSNP accession numbers, somehow ambiguous. This is for the first part of the error analysis. Then we also look at a subset of what seems to be, let's say, performing better for Varium. But still, we wanted to do this type of also qualitative analysis. And then we see that if we look at the average number of documents retrieved per query, so the use of the expansion is, of course, beneficiary. So we basically double or more than double the document can retrieve. Okay, so from 2,300 to more than 5,000. The number of queries with no results decrease significantly from 255 to 137, and the estimated precision decreased, but let's say remain relatively high. So 93% precision is regarded as relatively good by the curators. And this is also available. So what we could say is that so far so good. Variome is a high recurse search engine that can support curation with, okay, the result that are there. So about plus 20% recall and minus 43% silence. The next steps would be to use the supplementary data, which obviously are useful for such a task. So this is the last evaluation. So we try to see how much, what could be the impact of using this supplementary data index. The idea came from the 3-spot curators first, that clearly say that they occasionally look at the supplementary data, but also from results published by Jimeno Yepes and Verspur in 2014, which say that supplementary data are a gold mine for variants. So in that case, in this last set of experiments, we look at only this supplementary data. So to find supplementary data could be challenging. So you could try to take them all, but that would generate a lot of noise. And normally, when you work with search engine, then you would first build a search engine and then use this to search. So it's a chicken and eggs issue. So what we decided to do is just to use the indexes that we have locally, textual indexes, to fetch a subset of document out of which we are going to retrieve supplementary data, which then will be indexed. We also built a binary classifier, but I won't report on this now. So we process supplementary data such as, CSV, XLS with simple transformation tools. And for images, which are the largest part of this supplementary data, we use OCR, which are now indexed into CBILS. This is just to show you the distribution of this supplementary data. It's first images, second text, HTML, PDF, whatever, and then of course a lot of tabular data. So this is the evaluation. So now we use that as a literature triage system to support a curation at a time. The curation of one variant at a time, not for the VCF one. And you see that the average number of retrieved document is much higher, about twice larger. The number of queries returning 0 document really reduced. It's basically the silence disappeared. And if you look at the precision, we have a drop, about 20% drop of the performances, but still remain, let's say, manageable. So there are some small differences, which makes that probably the real precision is slightly higher than 75%, but I won't enter into detail here. So just to show you a few errors that we have. So sometimes there are let's say mismatches. That one at the bottom here between SLC 6A11 is actually a wrong mapping, which is R332 key from BRCA1, but in a different gene. So this is I would say a confusion. And the same happened with the images, of course. You can see here that, for instance, you have in the images, you have Q220 star, and this is transformed into Q220 percentage. Of course, the system is not able to correct this type of misspellings in a sense. In conclusion, so Variome is setting a new state of the art for variant search. And this is done by leveraging dirty and messy supplementary data, and by using intelligent search systems and text analytics. So this is maybe a broader take-home message. So FAIR is a very important, I would say, duty these days. And of course, we need to leverage the existing infrastructure and ecosystem like the one that exists. In parallel, we also need to establish stronger guidelines and standards. But of course, standards are always coming after the data is created. So I think it would still make sense to try to pay attention to supplementary data, which I think have been, let's say, somehow neglected in the past. So the system is now up and running. It has been designed and tested by curators from UniProt. It's piloted by clinicians, in particular, actually, anatomopathology, oncology, and hematology. It has been in production for now 4 months. And of course, if you would like to have more information about the detail of implementation for, in particular for VCF, which is a sensitive content, and so please feel free to contact me. These are the links to the different services that were shown, in particular Viomes. These are all the people that contributed to the systems, including people that take care about the legal part, like Valérie Barbier, including not only, but also the legal part. And these are the different supports that we received for this project. Thank you very much.
Thank you, Patrick, for a fascinating presentation. We have a couple of questions, which I will read out now. So question number 1, do you think societies and research funders and journals should encourage data sharing via repositories instead of supplementary data?
That's that is a very good question. So I think that we are investing a lot of resources into data management, and that is legitimate. However, my impression is that we are probably, these days, we are probably making a strong effort into standardizing and let's say curating metadata, and probably a bit less in this supplementary data. So actually there have been, I don't know in the US, but in Europe, there have been quite few grants dedicated to the management of supplementary data. And I know at least, only a couple of them at the European level, but it has remained relatively marginal. But the key question would be probably, should we reduce the investment into dbGaP and EGA and reallocate more resources there? I'm not sure. That is really something I'd like to debate, because we also need resources there. So yeah, but I still would like to see some initiative going that direction. Yeah.
So thank you very much. There are 2 more questions that I'm going to ask you to type in an answer to. But thank you for an excellent and thought-provoking presentation. I'm glad you've left off the resources, and we will now move on to our next speaker. So our next speaker is Dr. Joe McIntyre. Joe is a senior scientist and associate director at EMBL EBI Services with responsibility for data resources. Prior to that, she was team leader for literature services at EMBL EBI, running Europe PubMed Central, which we just heard talked about and used. It's the open access database of abstracts and full text articles for the life sciences. And Joe's primary focus has been on integration of the open access literature with life science data to support development of new ways to access and discover research information. So Joe, you're on.
All right, thank you. Hello, everyone. So I'm going to talk a little bit today about Europe PMC from sort of 2 perspectives. One is in its core service provision, which I think all curators search the literature, well, all scientists, especially curators, search the literature. And I wanted to highlight a couple of unique things about Europe PMC. And then secondly, I want to talk about some of the work we do, which is about building infrastructure to support the sharing of annotations, text-mined annotations in particular, which we hope that ultimately having a shared platform for these expertly extracted annotations will provide a good basis for helping to develop curation tools. So firstly, just a little bit about Europe PMC. You probably know all about our big brother or our big sister PMC in the USA. We partner with them, and without them, we would have a lot less content. But we do something slightly differently from PMC in the USA. And one of them is that we have recently started to index preprints across the scale, across many preprint servers. Europe PMC is funded by 35 now funders of life science research in Europe and related funders beyond. Basically, it serves as a repository for author-accepted manuscripts when the case is that the open access policies can't be complied with by any other way. And all of the content is available via the website, APIs, and by bulk download, at least as far as the license allows. So there's many ways to consume the content. And if you search Europe PMC, it'll look something like this. I couldn't resist looking for the number of AlphaFold papers. Extraordinary, nearly 1,000 papers already published on the basis of AlphaFold and the sharing of that data set of predicted structures. And you can see here classic sort of I'm not sure if you can see my my mouse, but classic sort of list of results. There's different sort by dates, and also categorized into research articles, reviews, preprints as primary filters. Something about how extensively you can use the content, and a little bit of date sorting. And of course, there's many more filters. These are just sort of some of the top ones. So I'll tell you a little bit about the preprints that we've indexed. So we've now indexed, not far off 500,000 preprints from 24 preprint servers and open research platforms. And indeed, if you just do it, I know it's not quite true because we don't know how many preprints are life sciences, but if you just count the number of preprints that were out in 2021 versus the number of PubMed records, almost 10% of PubMed records now have a preprint. If you look at it, the numbers are that comparative. So about one in 10 PubMed papers could have a preprint associated with them. So it's definitely a growth industry. And Patrick mentioned his shift of behavior over the last couple of years, and I'm sure we've all experienced that. One of which for us was that we started indexing COVID-19 preprints as JATS XML in full text, and so that's a full text data set for those that is available as far as possible for text mining and for searching. And from later on this year, on the back of that work, we're going to start indexing all the CC license preprints from those 35 funders that I mentioned and making those available in full text also. Finally, recently for curators, this was some feedback from the curator community at the EBI, because as you may or may not know, but where I work at the EBI has a large number of databases and a cohort of curators that really do scour the literature for facts to extract and put in various different life science research databases. And one of the things they wanted was to know, one of the things they've been thinking about is whether they should curate from preprints or not. That's a whole other discussion that would have been a fun one if we were in person. But definitely if they were thinking about curating preprints, then they want to know when that preprint has been peer reviewed and published in a journal. So we've set up recently alert services that you can sign up for to know when that event has happened. But all in all, preprints are definitely part of our ecosystem of papers that we can use. There are just, a large portion of go on to be published in peer-reviewed in journals. Many have been cited. A large portion have been cited, actually, over half, at least once. They've been claimed to ORCIDs. They have data attached. Some of them have open preprint peer reviews attached, and even some of them have lay summaries as well. So I think, the pandemic has really accelerated our means of publishing, and I think will change the game in the future. So another part of what Europe PMC has done historically is to really try and connect all the literature with the data, and we do this in a number of ways. One thing I want to mention here is we have a sort of process that mines many accession numbers from about 40 different databases on a daily basis. And this is quite useful for curators and data resources because they can then look to see which papers have cited accession numbers in their from their database. And sometimes, they find new papers that have made use of their data resource. And that's a useful thing for both enriching the data resource and showing funders that, how widely used the data resource is. Another part of what we do is this TextMind insights, which I'm going to come on to in a moment. But we also do this, and many data resources do this with us, where we have a sort of link out system for databases, so that when a paper has been curated in a database, we can reverse the link and point to the database. And this proves quite useful if you're looking for, papers to curate, and it might be useful to know that it has already been curated in a different database, either as a sort of assurance of quality, or that you perhaps would pass it over and look for something else to do. So it helps in triage. I wanted to talk about our PMC annotations platform. So of course we provide all this content, in different ways, but also what we really would like to do is enrich that content as far as possible. And this is to use in curation workflows. And this is a recognition that we cannot, no one team could do this. It's really should be a community effort where, we heard Patrick's talk about his expertise there, and that's certainly something we'd love to incorporate into the database. So basically we provide the content, text mining teams do their text mining. There's a submission system to submit the outputs of the text mining performed. And this platform has an API, which is consumed by the Europe PMC website, and we can re-show those annotations on the paper as highlights, and I'll show you that in a minute. But also, because it's an API, third parties can use them. So anyone can come and use all of the deposited annotations in the system. And because we've taken this sort of community approach, there are like a vast array of types of entities that have been mined. And here are some of the collaborators. And it's all done in the standard format, which is the web annotation data model. So in theory, these annotations can, cross-compare with other resources that have used this format as well. I'm blanking on the name actually. It's what is it? The Annotate the Web guys out in California. Perhaps someone could remind me of the name of that in a moment. But they use the same format. Something we've done quite recently, we've been trying to get out of the door, but we got distracted over the last year or two, is to use machine learning to improve the core annotations that the Europe PMC team provides. And to do this, we had to make a training set. And we've been in the process of making this training set on full text articles available. And very soon, it's already available. You can see the link here, but actually we're going to write a paper to highlight the availability of this training set. And basically we annotated 300 full text open access articles with genes, proteins, diseases, and organisms. It was quite difficult to think about which papers we should select out of the 6 or 7 million that are in PMC now to be the 300 in the training set, which of course was constrained by budget. As you know, these training sets is quite expensive. So we basically filtered by license to make sure that the data could be reused, and then we picked sort of representative articles based on body size, and that reduced the number quite significantly. And then we worked out how many, what the sort of frequency of annotation was for genes, proteins, organisms, and diseases. And we made 3 sort of bins of low for each one, which would be low frequency. It would be low occurrence, medium occurrence, and high frequency of occurrence of the different entities. So in total, this made 27 bins. And you can see on the graph below that we call these low, medium, low, high, low, medium, medium, and so on, which would represent, a set of articles that had low genes, proteins, low organisms, medium diseases, and so on. And then we took so when we had binned all those articles, you'll notice that the sharp-eyed among us will notice that the 27 bins here, there are only 26 in the graph, and that's because we removed the bin which was low genes, proteins, low organisms, and low diseases, because the information content in those was pretty low for our exercise. So for the remaining 26 bins, we sampled from them sort of proportionate to the size of the bin, and this is how many articles we took from each bin to make up the 300 papers. So this should be out in a paper very soon, and all the text miners who are listening might want to take a look at that, because there's only one other full text corpus that I know about, which is from Larry Hunter's team. Okay, so because we have this infrastructure, and because we've been doing some of these things for some time, we could be quite we could pivot quite quickly in the pandemic, and so we very quickly added the GISAID accession numbers to the accessions that we mine. And working with Patrick Ruch and his team, we also layered on top of the Europe PMC content, the COVOC ontology for COVID-19 terms. Another of our projects is the Open Targets project, and this is where two members of the Europe PMC Literature Services team, who are shown here, have been mining gene disease associations and pushing them into the Open Targets platform. Open Targets is a public-private partnership that uses human genetics and human genomics data to identify drug targets. So this is a pre-competitive platform that's completely open for anybody to use. And so we have had some ongoing work there to, I say, improve these gene disease associations, but also add clinical drugs. And the way that they do that is that Sharma and Santosh produce new outputs, and then the users of the Open Targets platform give feedback, and the algorithms are adjusted accordingly. So this has been a very helpful process, and of course all these annotations are also available in the platform. Another project I'd like to share, so this is sort of trying to demonstrate, not only doing something useful, but trying to demonstrate the use of a platform. And this is work from Marley Nasser, which is a project that's almost come to an end now. But she did a most fantastic job in mining microbiome terms from the literature to enrich data sets in Magnify, which is the metagenomics dataset database. And if anyone's ever looked at these Magnify, at these metagenomics data resources, they will have noticed that the metadata is in some cases extremely poor. And so the idea is to, and if you don't know the specifics of the biome, then if you want to do any cross comparison of data sets, it's going to be very hard to do, because you can't work out from what environment the data sets come from. So you can't really compare them properly. So she has enriched 16 entity types to make these kinds of experiments more possible. And I'll give you an example here of how it all fits in with the Europe PMC infrastructure. So here in the top left, you have a Magnify record of a study. And the title tells you something actually, but in terms of the sort of deep metadata applied, it's literally only has host associated human assigned to it. But when you find the paper that's linked to that record, which is possible to do via the European Nucleotide Archive, and you apply Marley's algorithm, you can then extract all kinds of useful information about the biome and also the methodology, which is also important. So here we learn that we're talking about a Japanese cohort, and you can't see on this list here, but you can see that we've got women, pregnant women, and so on, which is, it significantly enriches the word human. So if you wanted to, for example, do cross comparison of, well, pregnant women, or even just women, never mind pregnant women, then you could pull this data set out, whereas previously you could not. Okay, so finally, some future directions. Patrick mentioned Elixir, and we've been a big, this is a bioinformatics infrastructure in Europe to try and coordinate across data resources and services. And one of the things that's been happening recently is a call for implementation studies for curated data resources and to how to enrich them in a more scalable way. And 5 projects were funded. They all started, I think, in January this year. And of the 5 projects, 3 will collaborate with the Europe PMC infrastructure to push annotations into this system and share them more widely. And so we should have extra annotations in the next year or two of transcription factor, target gene interactions, phenotypic data, and also lipid pathway data. And finally, there is a new program of fellowships at EMBL called ARISE, which offer 3-year fellowships for either if you're trained scientifically or if you're trained perhaps in technology to come and study some advanced form of service provision. So this would be, the cutting edge of some of the data resources at the EBI, or potentially instrumentation innovation in, for example, for microscopes and things like that. So what we're fortunate to have having an ARISE fellow starting this year who'll be looking at how we can use the literature in a more scalable way for database curation, in particular working with the INTACT database, the protein-protein interaction database. So on that note, I would just like to thank all of the collaborators who've donated and continue to supply annotations into the Europe PMC system, on the funders of those systems. And I have some rather blurry pictures of some of the people who've done the work and that I presented, but also I'd like to take the opportunity to introduce you to Melissa Harrison, who's a new team leader for literature services at the EBI. She started, which was my old job. And it was my job when I accepted to do this workshop. It's been delayed. So Melissa is now the new team leader of literature services, and she started in November last year. So welcome, Melissa, and I hope she'll be taking this work forward in the future. Thank you.
Thank you very much, Joe. That was fascinating, and you already have a lot of questions. I'm going to grab one question from the chat if I can find it. From Jiang Lu. For the full text data set, where do the gold standard annotations come from?
Well, we employed a team of curators, basically. So they're human curated. I should have said that. They're triple blinded. It's all in the paper. I can let you know about it if you like.
Wonderful. Yeah, if you can put pointers in the chat, that would be.
Yeah, I will. Can have all the audience see the chat as well? Which chat?
Yes, the audience can see the chat.
Okay.
Here, another question. How does the team handle noise from working with the full text articles versus title and abstract? Do they focus only on certain parts of the full text, and how do they handle tables?
Yeah, that's a good question. Well, I mean, you can look at term frequency and so on, and that helps you parse out the noise to some extent. And you can also focus on sections. We have a standard sort of section tagger. So in general, the introduction can be quite noisy, and you're better off focusing for most things on the results. Other things for the methods, but of course some papers don't have nicely divided articles like that, so it always will be a little noisier. And yes, tables are really hard. I won't deny it.
Okay,
they don't work in sentences.
One last question from Yan Li. Whoops, just scrolled up my view here. The figure shows article count regarding low, medium, high protein gene disease. Could you provide some insight about the articles in the low, low, low bin in the context of innovation, breakthrough, and new research areas?
Oh gosh, what a question. I mean, not off the top of my head is the honest answer.
Okay.
It would be good. It would be interesting to. It would be interesting to look at the low, low, low bin. I think it might be that our organism, disease, gene, protein interests, if you like, are quite molecular, and PubMed Center is full of not just sort of molecular biology. A lot of it is quite clinical, and in that case, it might be that. That those were the low, low, low papers, I would think, or comments and things like that, letters to the editor. But yeah, it'll be interesting to look and see what kinds of articles are in there.
Okay, and I know I need to stop, but there's a really interesting question from Kimberly Van Auken. Do you use results of preprint indexing to feed back potential errors to publishers that could be fixed prior to publication? Just
no. But,
okay.
But that's a good idea. That would be tough. I mean, I certainly get on my high horse a bit about in some of the preprints, the handling of data links is quite awful, with
Okay.
the XXX written instead of accession numbers and things. So I think really to feed back to the preprint servers to sort of polish up on some of their processes might be the way to go there. But we don't really know at the time when it's a preprint where it's going to be published. So it's actually strangely difficult to make these links, which is why we're quite proud of the alert system that says when and where a paper has been published in a journal.
Great. Well, thank you very much. I'm going to move on to our next speaker, but and also you talked about wanting to discuss some of these things further. There will be a panel on curation at scale in the time of pandemics tomorrow. So encourage everyone else to attend that. Okay, so our next speaker is Professor Alfonso Valencia. He is the director of the Life Sciences Department at the Barcelona Supercomputing Center and also the Spanish National Bioinformatics Institute. His research is focused on the study of biomedical systems with computational biology and bioinformatics approaches. He is a founding member of ISCB. He served as the president of ISCB for between 2015 and 2018, and he is also a founder of BioCreative. So over to you, Alfonso.
Thanks, Lynette. Thanks very much for the invitation. And good to be virtually here. So let's see if I can share my screen. Yep. So first, so what I thought for these sort of 20 minutes is to tell you first a little bit of background why we are interested in a number of problems related with natural language processing in medical records. We are doing this in Spanish, so you are going to get efforts and examples in natural language processing in Spanish. I will tell you about the two systems that we are developing for the two language models for Spanish and Catalan that we are developing, and then how we apply this to medical text. And I have a few minutes more, and then I will tell you about the benchmarking effort of systems in Spanish and how we foresee the organization of these benchmarking efforts in a more sustainable and automatic way. So first, let me introduce you the place I work for the last 4 years, the Barcelona Supercomputing Center. Obviously, it's a supercomputing center because we have a pretty large computer. We're installing a new one that will be one of the largest computers in the world at the end of this year. And my department, essentially, I will tell you about two of the things that my department is doing. One is natural language processing, and the other one, a little bit about what we do in terms of infrastructures for biological data. So my motivation, and let's give you the context, is we are starting now the Spanish medical genomic or personalized medicine project. So this means organizing the system for introducing genomics into the medical practice, and doing this systematically, as many of the other countries are doing. My example here is from the Swiss medical genomics or from the European health data space. We are responsible of the handling of the data for the Spanish personalized medicine project, what in practice means being able to install the infrastructure. We can do that. Being able to handle the genomic data, we can do that, but also means being able to handle the phenotypic data, essentially the information from the medical records that is essential for the interpretation of the genomic information. So we end up working in this area of medical information, extracting information from medical records, and thinking how to apply natural language processing methods in this area. I'm not going to tell you about the difficulties of operating with medical information. I think this is a well-known problem that has all kinds of difficulties. The data is difficult to access, it's difficult to discover what is in the data. There are physical problems of accessibility with the data distributed in hospitals in different databases and systems. There are obviously problems with the data that is protected with different protection systems and difficult to get to. There are problems aggregating this data, even inside a single hospital. There are a lot of problems making the operations of data extractions automatic. And then there are huge problems on semantic interoperability. The examples here are from two very well-organized Spanish hospitals talking about COVID data. The two numbers are the number of, they put together the data for the COVID cases, and the two numbers for each one of the modules, outcome, for example, the 7 is then after solving the problems of syntactic interoperability, is the number of cases or items that they match and they have the same meaning. And the 131 is the number of cases that they need to make a transformation to be able to match the meaning of between the two hospitals. So a lot of work just to match data from two hospitals, even if they are two very well aligned ones and has been working together for years. So again, a lot of problems in this area, not one, just to give you the context of something that you probably know about the difficulties of working with this type of data. So we thought that maybe part of the solution, or at least we can do some contribution to in this area, using natural language processing approaches to the Spanish text. And in this case, the two things that I'm going to tell you is how we do this, how we go about developing models. And this is the work of Marta, and then how we apply this to medical records in collaboration with the Spanish hospitals. And this is the work of the group of Martin Kralinger. For the Spanish and Catalan language models, they are called, Aina is the model for Catalan, Maria is the model for Spanish. The model for Spanish is pretty large, it's the largest model for Spanish as far as we know. It's a GPT-2 model. Built with a large corpus from the Spanish National Library. Aina is smaller. We are now in a campaign to obtain more text for Catalan, and so to do this in not only in the written language, but also in the spoken language. And the current model has 1.7 thousand million tokens. And then in parallel, we are developing this other corpus, biomedical corpus. This is one of the reasons why I showed you at the beginning our supercomputer. This is Marta and Maite in the location of our supercomputer. It's a pretty impressive location, I would say. And this has become a challenge for our computational capacity. Indeed, to develop a larger model than the one we have now for Spanish, we have to wait for the new computer or new Mare Nostrum 5 that will be ready at the end of this year, because with the current one, we don't have enough capacity to calculate the larger model. Just to give you, sorry, because this is in Spanish, because well, I used to give this talk about the Spanish models in Spanish. So the text we are using is from the National Library. They are collecting all the web in Spanish, and this is the collection of all the web of Spanish in Spain for the last 10 years, so it's a pretty large text collection. The calculation here is that it's 40% of the size of Spotify if you count all the download. Obviously, you have to clean the data, move this from the web text to a text that you can use. Cleaning the data, as you can imagine, has been even more complicated than calculating the models. And then we spend more time in the curation of the corpus than anything else. And then we have trained these models that training a single model is taking the equivalent to 200,000 hours of computing. Obviously, we have trained a number of models. So in total, the Spanish model has consumed something like 20 million hours of CPU. Now the model can be used for many things, and we are exploiting this in collaboration with many different groups for tasks that are anything that you can imagine: generating text, classifying text, making summaries, adapting text, etc., etc. In this context, obviously, the more interesting applications for us are the ones into the biomedical domain. So this is natural language processing in Spanish. As you may realize, Spanish is a language spoken by almost 800 million people in the world, but is not that well represented in terms of natural language processing, certainly not as represented as English. The number of resources there are a number of resources, including a lot of publications in Spanish, particularly in the medical domain. There are journals and collections of papers in Spanish. There are very interesting collections of clinical trials, a full database, and a collection of patents, clinical practice guidelines, social media, and also the research projects. So we are collecting all these sort of standard data sources as the corpus for training these biomedical natural language processing systems in Spanish. Problems that we are finding, there are a lot of Anglicisms, a lot of English word or English adapted words in the Spanish text. In the case of the medical records, at least the ones in Catalonia, there is a mixture of Spanish and Catalan with no logic. They can be mixed in any way. There are, as in other medical texts, there are a lot of abbreviations, acronyms that complicate the operations and require some specific treatment. Medical records are written in this type of telegraphic language that is really not very appropriate for natural language processing. One specific problem in Spain, we have a lot of localism. So we have a lot of doctors and nurses from Latin American countries, and they use a different terminology. And things can be very misleading. The same word may mean different things in different versions of Spanish, and that is again an additional complication. And in other cases of medical text, negation is an essential component. And then there are a lot of variability between different types of documents, depending on the medical specialties. So some of the things are common to all the medical records, some of them are very specific of Spain, I would say. So we have developed a full pipeline. It's a collaboration with a hospital in Barcelona, hospital clinic, a hospital in Madrid, 12 de Octubre, and another one in Seville in the south, Virgen del Rocío. So, like in other cases, annotation guidelines, creating gold standards, training the models with this gold standard, and then with the help of the clinicians, going through a number of rounds of manual curation and retraining, training new models, and then annotating documents. This part of the is called here the clinician in the loop is essential, is involving the experts in helping us to refine the pre-annotated text and providing additional annotations, and then going through all these a number of times. And this is a significant effort, and this is the case with the clinic. These are more than 90 sessions of 1.5 hours of training, consolidating, and scaling up the system. We divided the experts in teams of 2 experts, 5 teams with 2 experts plus 2 linguists, and in total they have been working in annotation tasks for 100 hours of the clinicians. There is a collection of 400 documents that has been annotated following this, and this is including the annotations of diseases, drugs, procedures, pathogens, symptoms, and professions. And then with this, we have built models that are now analyzing these type of entities and extracting these type of entities from the text. So the number of texts of different sources from 2 of these hospitals is the order of thousands for different type of medical texts, from radiology, when they are moved from one part of the hospital to another one, from when they are released from the hospital, or the clinical trajectory. And this is one example. This is a piece of a text, just to give you an idea of the level we are able to achieve. There are diseases that are identified. There are different type of dates from when the proof was made, when something happened 15 years ago. We are detecting a number of negations of different types, with different type of negations. We are detecting uncertainties. Indicaria is not clear, that maybe is a potential sentence. We are detecting drugs, and we are detecting symptoms. In this case, this is a good case. We are finding almost everything that is in the, being able to assign almost everything that is in the text. The systems are working. I'm not going to go into details, but are working very well for things that we know that are easier, finding pathogens or finding names of diseases. They are more difficult. There are things that are more difficult, are procedures and symptoms. Symptoms in particular is a very difficult, ill-defined category. Interestingly enough, even if the texts are a mixture of Spanish and Catalan, doesn't make a difference. The system we're not trained specifically to distinguish Spanish and Catalan. They are working equally well in the two languages. The two of them are similar but different. And they're also working, I would say, quite well in detecting negations to a high recall and F1 score, but also the other qualifiers of relative, the terms of relative impact of different things. So we are, well, this is as far as we managed to go with this type of approaches in collaboration with the medical doctors. I want to highlight that this is only possible because the medical doctors took this very seriously, and they put a lot of effort in the annotations of the text for training the system. So what I think is one of the lessons that we learn from that is how to engage with them and how to develop this in a collaborative way. As you know, we have been, Lynette already mentioned Biocreative. We have been always very fond of organizing these shared tasks and challenges. These are the ones in Spanish, same than the others. Prepare a relevant text, give this called a standard for a given area, give this to a number of teams for them to prepare the preliminary system, develop the systems, and then have final systems that in our case are servers. So we do this automatically from servers. These are the challenges that we have been organizing the last three years, and some of them are for this year, in medical abbreviations, semantic indexing, medical documents, detecting clinical tumor morphology, clinical coding, ICD-10 codes, detecting professions and occupations in medical text, and in social media. So I'm not going to go to all the details, just to highlight. In this one is the one of tumor morphology. This is organized with the professional society of pathologists, and there has been 121 automatic systems participating. And of all that, we are obtaining 7 systems that are open and can be used openly. So I think it's it's a remarkable success for a medical task in Spanish to get to 120 systems participating, with results that you have on the lower right corner that I would say are pretty good in terms of the accuracy of the systems in being able to detect the morphology of the tumors and normalize this to a given ontology. These are the ones that we are still preparing, or we are still organizing for this year. So if you are interested, you are most welcome. Diseases, species and pathogens, and automatizing the translations in the medical domain. The last thing I wanted to say is that all this is a lot of work to develop these tasks, and we know this for Biocreative, and in a sense it's okay, you do it once and you assess how is the state of the field, but it will be better to have this done in an automatic and sustainable system. So we have been developing for the last few years this open events as a system to do this type of evaluation systematically. So essentially it's engaging with communities, trying to get from them a collaboration to what is the reference data set, how they want to evaluate their methods, and then do these evaluations automatically and systematically. So we have already a number of communities that are working this way. So we have their data, we have their standards of evaluation, and we are continuously or periodically evaluating their methods. So this can be evaluations of specific individual software systems or given software at the technical level. Is the system up and running? Is what is the uptime? Is accessible? Is open? Etc. Etc. Or this can be visualization. These are visualization for a given community. The Quest for Orthologs. Visualizing the results, and this is a service to the community in a way that they can compare different software, different methods, what is similar to what, or systems for
About a minute to wrap up.
running tools or doing this benchmarking systematically from beginning to end, installing the software, running it, and analyzing this in a continuous way. So I really believe very much in the importance of being able to do this scientific benchmarking systematically to complement the campaigns that are done for a given subject at a given moment. So I told you a little bit about semantic interoperability and medical records as a big problem, the models that we are developing in Spanish and Catalan, applications to medical records, mining medical records, the specific challenge in Spanish, the task challenge in Spanish, and this idea of systematic benchmarking. If I want to have my summary, what we learned from all that is the importance of gaining the trust of the communities. In this case, the medical doctors or the communities engaging in the benchmarking or with the different challenges. And engaging with the medical doctor was easy or relatively easy if they understand why we are doing that for, and they see that this will be a tool that will be installed for them to use it in a given future. Now we are installing part of the system in the hospital, and that's another adventure. But I think that's part of the contract, part of the way of gaining the communities that they see a tangible benefit of this collaboration. From the shared tasks, I already said that they are very important for gaining input from the community, for assessing things, for getting data set, but also I think that it's very important that we are able to do systematic evaluation and that these tasks are linked to the position of the software in a way that can be used by the community. And this has been one of the struggles of all the benchmarking effort, not just to produce results, but also to, at the end of the day, produce software that the communities, the users, accessible to the users. So I finish with that. I just want to mention that essentially this is the work of Martin on the text mining, biomedical text mining, Marta team on the development of the large language models, and Salva Capella on this benchmarking, and his team on the benchmarking effort. Thank you.
Thank you very much. Wow, you covered a lot of ground. While I wait to see if there are questions coming in, I'm going to ask you a question. I missed whether the medical record data that you've gathered is de-identified and shareable outside the particular hospitals that have provided the data.
Not at this point. The one we are using now is inside the hospital. So the systems are running inside the hospital with no anonymized data. The project is to anonymize the data and to make accessible at least a part of that, not the full thing, but at least the course of the diseases or the course of the course part of the medical records, because the other ones are more complicated for legal reasons to make accessible.
A question from, let's see, a quick question from Jiang. Are there things we can learn from Spanish language processing for English? I would add to that, one of the ways to get even more people to work on Spanish is if you could make the data sets available, because these are, as you pointed out, extremely rich and hard to get data sets. So
I think that we have already plenty of non-Spanish speakers working on the competitions that Martin Krellinger is organizing. Because at the end of the day, you just train systems, so you don't really need to understand the Spanish. So I think it's a very good. So these 100 systems are not all of them from Spanish teams at all. And the data, all the data for all these competitions is accessible. Again, the hard bit is to make the medical records accessible, and this is what we are working to. And that goes back to the previous questions of anonymization.
okay, thank you very much. There are more questions, but I think we need to move on. So there's some questions in the chat. And our final speaker of the session is Andrew Su. Andrew is a professor at the Scripps Research Institute. His lab's research focuses on building and applying bioinformatics infrastructure for biomedical discovery. And Andrew has a particular interest and a long track record in applying crowdsourcing to organizing biomedical information. So Andrew, over to you. And we see your slides.
Is
So
he
you
muted?
can-
Andrew, I'm sorry, it appears we're not hearing your audio and you are unmuted. We may need you to reconnect your audio. Can you? Yeah, I'm afraid we're not hearing anything. If you can go down to your audio settings at the bottom and check your audio settings.
Okay, testing 1, 2, 3.
There you are. Good, thank you.
Sorry for the debugging. Okay, and can you see my screen?
Yes.
Okay, fantastic. So thank you to Lynette and organizers for organizing. Really enjoying this meeting and looking forward to more. So since we're at the NLM Curation at Scale workshop, I thought I'd set the stage by really, thinking about what we're trying to scale to. So my lab is very interested in building these large heterogeneous networks. I'm showing one here that was created by Daniel Himmelstein many years ago, which he calls HedioNet. This is a knowledge network that joins, in which the nodes are some of the classes you see on the slide here: genes, drugs, diseases, side effects, pharmacological classes, and the edges are the relationships that join them. So HedioNet has, on the scale of tens of thousands of nodes, millions of relationships, sort of later iterations that he and others have built, including our group, maybe are one order of magnitude larger than this. These attempt to capture sort of the relationships that exist within the biomedical literature. So, what is the substrate behind these large heterogeneous networks? It is the literature itself, of course. Let's see. It is the literature, right? And as I think we can all appreciate, the literature is growing at an exponential rate. This just shows the number of articles every year, right? In the latest iterations, latest years, the number of articles per year is much higher than even 1 million per year, which roughly averages out to one new article every 25 seconds or so. So clearly, the scale here is quite substantial. And so how do we scale to this, right? Right now, the process of going from that massive literature into these heterogeneous networks is based on professional manual biocuration. It is people who are reading literature, structuring that information in terms of biomedical ontologies and other structured data. And then that is the data that we integrate into these het nets. But what we know, of course, is that the professional manual biocuration doesn't scale with the growth of literature. Alison Lister put it under the nice catchy tagline yesterday that curation doesn't follow Moore's law. And I think that's obvious. And so the disconnect between that scale of the growth of literature and sort of in our professional manual biocuration bandwidth is the gap that we're trying to address here. So just what I said. So there's a couple different strategies we can imagine to bridge this gap. We've heard a lot about text mining as being an important tool, and I'm going to hear more, and that clearly is part of the solution. But I want to suggest that there's another quantity that actually does scale roughly with that growth in the literature, and that is the growth of the biomedical workforce, right? We have much more literature because we're doing more science, and we're doing more science because we have lots of people doing science. And so the fundamental question is, how do we engage those individuals in a way that might allow us to better scale with the growth in the literature? This has fall under the umbrella of community curation. And relative to other automated methods discussed in this session and elsewhere in this meeting, we can think of community curation as a type of human computation, which is sort of a class of computation in which tasks are distributed among large communities of humans, and together with computing resources can be used to tackle large computing challenges. Okay, so community curation, of course, is not terribly new, although enthusiasm for it has steadily grown over the years. And so there are a couple, again, different strategies for how one might pursue community curation. One strategy might be to target communities of domain experts and domain consumers. And then within that community, try to build that culture and the incentives for data contributions. I'll highlight the great work that we heard yesterday from Kimberly Van Auken around Wormbase's efforts to do community curation. Other model organism databases and other databases from Flybase and Uniprot and so on and so forth have also looked at community curation, and I think have made some fantastic results. I think it is fair to say that this step number 2 is a challenge, right? How do you incentivize the domain experts who are already at, for example, your mod, to then also turn around and contribute is something that I think is an interesting and active area of work. I think the complementary way of going here is to target a community, an existing community of open data contributors, and then identify individuals and groups within that community with the appropriate domain expertise. And this is where we've really started to focus on using Wikidata. So for those who aren't familiar with Wikidata, Wikidata is a sister project to Wikipedia. So Wikipedia is to text what Wikidata is to data. So it's a community edited knowledge graph of structured data. And the goal of our project, which we put under the umbrella of something we call the GeneWiki, is to really cultivate the biomedical corner of Wikidata and use that as a method to structure information. So Wikidata as a whole has about 97 million items, different entities, obviously spanning not only biomedical entities, but movies and actors and geographic places and so on and so forth. But to make the point that it is an active community of open data contributors, right? Wikidata in the past 10 years or so has accumulated 1.6 billion edits, now has about 6 billion statements on these 97 million items, and has about 20,000 active editors. And so the question is, can we use this resource as a tool for structuring biomedical data. I'll say that this GeneWiki project started in the context of actually Wikipedia. And so, I'm just going to highlight one particular snippet out of the Wikipedia article for this protein relin. And it says the expression of relin has been found to be significantly lower in schizophrenia and psychotic bipolar disorder, which is great in free text, but of course that isn't a structured bit of information. Wikidata provides the infrastructure by which you could structure a free text assertion like that. This is the corresponding page for relin in Wikidata. This is simply a web page, but let me just highlight what it is that everything on Wikidata is essentially a triple. Relin is a subclass of a protein. Relin physically interacts with VLDL receptor and amyloid beta A4, so on and so forth. Relin has decreased expression in schizophrenia and bipolar disorder. Again, going beyond the web page is that all of these used unique identifiers, so we can really move beyond obviously just the free text. And then this is not only accessible in a web page form, but of course in structured data formats like this JSON output that is available for computational query and analysis. I'll highlight as well that the data model behind Wikidata also incorporates qualifiers. So you can say qualifiers like a determination method and references. The referencing model has changed a little bit beyond a naked PMID, but referencing is sort of baked into Wikidata. So after the last talk, I couldn't, Afonso Valencia's beautiful talk, I couldn't help but throw in this one slide, just emphasizing that Wikidata is meant to be multilingual at its core. And so, for example, type 2 diabetes, you can see, for example, there's probably 50 some odd languages there in which you have the translation of type 2 diabetes. So that might be a useful resource for the text miner. And it's something that we've talked about with Lynn Schrimmel about automated methods of, for example, creating multilingual versions of biomedical ontologies. But nevertheless, if we focus for a second on just sort of the molecular knowledge graph that we've accumulated. So this clearly is a busy slide, but I'll zoom in a little bit just so you can see our core sort of part of the graph relates diseases. So we have about 17,000 diseases in Wikidata, 1 million genes among many organisms. And then down over here, chemical compounds. So this is sort of our central axis of how diseases relate to genes, relate to chemical compounds. And then around that, we can see the subset of compounds that are medications. We can look at anatomical structures. We can look at various pharmacological actions, taxa, and so on and so forth. So this is sort of a class level diagram of where we're at. And so I'm summarizing a huge amount of work to say that we've developed computational tools to import and synchronize data from many structured resources, including some of the ones over here, into Wikidata and to keep that up to date. So I think we in some of our published work, we demonstrated sort of useful integrative queries and integrative analyses, mining this biomedical subset of Wikidata. But I'll say that this knowledge graph, where we're integrating structured resources, is really a means to an end, right? If we were purely just integrating these large data resources, there are other efforts and other ways to do this outside of Wikidata. The point of bringing into Wikidata is to seed community contribution. And so now that's the question of, well, what do we mean about in terms of seeding community contribution within the context of Wikidata? So far we have imported data from large data resources. The first community I think we find is that the community of other data integrators. So sort of in our work, we were loading lots of chemical annotation data, and suddenly we started seeing lots of data associated with chemical toxicology and sort of the phenotypes associated with environmental exposures. And it turns out that there was another person within the EPA who was loading a lot of EPA data. And I thought that was a really nice bit of anecdotal data about how our data integration data efforts sort of meshed with things at the boundaries where we might not have identified them initially, but the stone soup analogy, if we all put this in one pot, then we can see the interfaces with other related domain areas. A big one, right, the one that we focus a lot on is how do we engage the community of subject matter experts? The domain experts who know, oh, I know gene X is related to disease Y according to this evidence. How do we get that into Wikidata? And of course, anybody can edit Wikidata, but the interfaces by which we engage subject matter experts oftentimes doesn't go straight through the Wikidata interface. And so we think a lot also about creating interfaces that are targeted towards specific domain experts. So this is one example for Clambase, which we created in collaboration with Kevin Hybisky, focused on the chlamydia research community. And so they were very interested in tracking things like genetic mutants and gene expression measurements and host pathogen interactions and so on and so forth. And so what we did is we created essentially a mini mod type portal. And it also incorporated the ability for people to add annotations. So for example, for a particular protein, we created these little wizards where people could add molecular functions or other Go terms to these various proteins. But what was completely abstracted from the user was the fact that we were both drawing data from and writing to Wikidata. So that not only were these annotations then visible within Clambase, but they would also get pushed back to Wikidata so it could be mined and accessed in a larger knowledge graph of information. And so Clambase was actually a generalization of a platform called Wikigenomes, which essentially could create that same interface for essentially any organism. But we're also thinking about other applications that essentially sit on top of Wikidata as a way to better engage with subject matter experts. And then the last thing I'll mention in terms of ways of tapping into the community is how do we engage with the community of data modelers, and here I'll point to our collaboration with Lynn Schrimmel and disease ontology as one example of, I think, where this worked really successfully. So we originally, again, loaded lots of data from disease ontology, and we put it into Wikidata. And what we found is that disease ontology, of course, being the authoritative source that we reference, but we found that members of the Wikidata community were doing two types of edits to those items. First, adding cross-references, and then also rearranging some of the structure around the diseases themselves. And so what we did is we actually fed that back to disease ontology curators for consideration of reincorporation into disease ontology, the official curated resource. And we did that through a variety of means, including CSV templates, robot files for people who use those. We did GitHub pull requests, and we explored a variety of different mechanisms that could work for disease ontology as well as other resources. And what we found is that the addition of cross-references to other identifier systems were accepted at a very high rate. Changes in ontology structure often weren't sort of immediately accepted whole as is, but it did prompt sort of investigation and sort of larger scale reorganization of different parts of the ontology. And so I think overall, this idea of feeding back the community edits from Wikidata back into these official curated resources turned out to be a nice strategy that leveraged this existing relationship and collaboration we had with the disease ontology developers. I'll briefly note that actually we weren't involved with this initiative, but the Wikidata community did mobilize very quickly for COVID-19. A wiki project around COVID-19 came into fruition in March of 2020, led by Tiago Liliana, whose slide I'm showing here, but really organized in this, again, this community platform information on case rates and death rates and other epidemiological data, scholarly literature, and also some of the policies and containment measures. So I think this was a nice example of how the Wikidata community, being a community of open data scientists, very quickly can pivot. So I'll end just by sharing a few perspectives in terms of what I think we've learned from Wikidata-based community curation. The first is that, Wikidata does have a large and active community of contributors, and they've come with diverse expertise. We have people who come in from software engineering and bot development, to ontologists, to folks who look at semantic web, to people who have particular domain expertise in many different areas. And so I think it is a useful community to tap into. I'll say that the data modeling process, when you're trying to data model by community consensus, it's a messy process. But I think typically it converges towards something that works for everybody. I'll say that partnerships with the owners or curators of authoritative resources was really critical. The last piece I mentioned in terms of the collaboration with Lynn Schrimmel, because I think that both in terms of how we model data within Wikidata and also what we can do when Wikidata sort of edits come in that conflict with the or are new relative to the authoritative resources. I think it's important to have that partnership to figure out what to do with those data. And lastly, I think we continue to think that data licensing is an important and still underappreciated issue among many biomedical data providers. Many people have hit on this issue. Melissa Handel clearly has been, who spoke yesterday, has really led the charge on a lot of these issues. But I still think, when we talk about inclusion of biomedical resources in Wikidata, a lot of people don't appreciate the data licensing issues that are involved. Wikidata did choose a CC0 license, so everything going into Wikidata has to be CC0. And I think when people think about the issues and understand them, a lot of times people are willing to relicense their data. So that happened with disease ontology, with Wikipathways, Reactome, Complex Portal, Civic, right? There's a number of resources that relicense their data in part based on our discussions with them in terms of the pros and cons of CC0. But I think that's still an area where biomedical resources could stand to look at these issues a little closer.
Andrew, we're about out of time. So.
Great, fantastic. I'll just end by saying that many people were involved, and I've highlighted them here, and I'm happy to take any questions.
Thank you very much. We'll do one question, and then I think we have to give everybody a short break before the next session. Question from Mike Livstone: if the community is to provide structured metadata, how do you train community members in the data model and enforce compliance?
If the community is going to provide data, right? Okay, so how do you enforce data compliance? Well, in truth, right? I mean, people can make any edit they want, right? But again, in the same Wikipedia model, where when in large data communities, essentially you have many eyes make all bugs shallow, essentially. So if somebody models data in a different way than the majority of Wikidata, then I think that just naturally gets corrected based on the number of people who are looking at Wikidata content. And so again, it's sort of this idea of at any given moment in time, Wikidata is clearly imperfect. But the argument is that it asymptotically approaches some sort of community consensus, with the understanding, of course, that community consensus can change over time too.
Okay, thank you very much, and thanks to all the speakers in the session. And I will turn it back to, I think we're on break now for the next, before the next session. So thank you.